Hello, we have Professor Javier Cabrera from Rutgers University today. Hello, Professor. How are you doing and how is this conference going on?
So this conference going very well. Today was the first day, and we had the opportunity to enjoy the beautiful Chennai and surroundings.
Okay. Okay. So, as I see, your topic is clinical study design using Hermite and propensity scores, external control arms, RWD, right? So, in that scenario, when randomized controlled trials are not feasible, how do you see real world data based external control arms reshaping clinical study design?
Well, the thing the current state of the data is evolving, becoming better and better every year, right? I mean, the data is collected by these companies, and is a is growing as bigger and better, right?
Right.
So which means that in the future, right, we expect that the real world data will be more and more useful in clinical studies because it is there are different points. One point is the issue about you have a new treatment, you have a patient of cancer, right? Why should you give it in the placebo, right? There is an ethical concern, right? The second issue is the issue of cost. The clinical trials, cost sometimes $1 billion. If you could reduce the cost to half, it would be fantastic. No?
And could you explain how the natural Hermite and propensity score indices improve reliability in comparing heterogeneous populations?
Yeah, so that's essentially the point. These are indices that measure the difference between the treatment cohort and the control cohort, right?
Yeah, yeah.
And of course, if we can make them identical, it will solve the problem, right? So it will eliminate much of the heterogeneity that existed by focusing in these groups that are identical, so we can estimate very accurately the treatment effect.
Okay, okay. My next question is, how does your work address the challenge of data heterogeneity in real world data, especially in regulatory contexts?
Well, so basically, our work tries to match the population of controls with the population of treated patients, right? The real world data cannot be used as treatment because, of
Correct.
course, it's not it's not new, right?
Right.
But they can be used as control by matching by using these indices that we propose. Of course, there is also other factors like, for example, the temporal issue, like real world data may be older or younger, right, depending on where it was collected, right? And because of older data may be different than the current data, right?
Right.
There's also a time component that can be incorporated, and this is what part of what we do as a learning, right?
Right.
We evolve the model to try to take this temporal component and use it in the formation of the new model, right?
Okay. Okay. And in animal studies, what advantages does your approach offer for reducing the number of animals needed and improving statistical power?
So the whole point is that in animal studies, you normally have the population of subjects already collected, right? It's not over time. You get
Right.
a box, you may get a cage with 100 mice, right?
Right.
And you need to split them into the groups for the different treatment arms, right? So in that case, our indices make the split optimal, so that the power of the test that you perform is going to be maximized,
Okay.
and as a result, it would imply that you could use fewer animals than if you were just using regular randomization.
Okay. Okay. And what are the implications of these methods for accelerating drug development timelines and reducing costs?
Okay, so mainly this is about reducing cost,
Yeah.
right? And of course, also accelerates the clinical studies, because if you, instead of say you need 200 treated patients and 200 controls, so we remove the 200 controls, right? then you only need to focus on 200 treated patients, right? So it takes half of the time to perform the study, and then it costs half of the money, right?
Okay.
So in both ways, it helps.
Okay. Okay. Great. And how do you see the adoption of these advanced statistical methods evolving in industry practice over the next few years?
Yes, I mean, of course, these things go slowly, right? Because first of all, we try to do like rare disease trials, right? That generally have few patients. Then also, we try to do trials where they use a small number of controls because of ethical questions. So you start with those collection of trials, and then if you are successful and the things look good, then you can expand, no?
Okay.
And that's how we will move forward.
Okay. Great. And are there particular therapeutic areas or types of studies where you proposed indices are especially beneficial?
Yeah, to start the cancer studies or rare disease studies, right?
Okay.
Where really it's very important the speed and the money, and also there are ethical questions, right?
Mm-hmm.
I think that's where we want to start.
Okay, that's really great, Professor. And yeah, it already started raining, I think. So thank you so much